right, our second day of working with Chapter 4, we're gonna kind of go back through everything we already did. Not in as much detail, but really want to make sure you understand. So if there's any part of it that doesn't make sense, now's the time to let me know. Remember, I told you this is probably the most important chapter we're going to do this whole semester. So I need you to really understand this stuff in depth. Okay? We started with what was the difference between a prokaryote and a eukaryote. I didn't really spend time on this slide last time. I just told you the basic sentence, the basic definition difference. What's the difference between a prokaryote and a eukaryote? Prokaryotes do not have what we call membrane-bound organelles. Prokaryotes do have organelles. They do have a lot of stuff on the inside of them. They just don't have membranes around those organelles. Okay? All the other things on this slide are just kind of some things to help you as you begin to study. We go through all of them as we go through the notes. Okay? So here was our prokaryotic cell that we started working on last time. And we started with some of the external structures. So to go back through those while we're looking at the picture, it's not on the picture, but we talked about the glycocalyx. Do you guys remember what the glycocalyx function was? Not secretion. It makes it sticky, so it can be a slimy layer and it can help it attach, or it can be more organized and it can be a capsule, which helps it stay alive. It makes it harder for something to kill that cell because it's got a nice little protective coating on it. Okay, very good. Then we went a little further and we started looking at the appendages of the prokaryotic cell. We went over the flagella. What is the function of a prokaryotic flagella? Movement. It allows the cell to move. There were different arrangements of the flagella. If I asked you what peritricious means, what would you tell me? Multiple, many flagella all over the surface of the cell. What about amphitricious? Opposite ends. So you'd have a cell with one flagella on one end and then another flagella on the opposite end. Lophotricious. More than one in one specific area. Because peri means more than one too, right? So you got to add that to that definition. We talked about how a flagella has a basal body, a hook, and a filament, right? So it's kind of in a fixed position. All that flagella can do is rotate clockwise or counterclockwise. One of your classmates noticed that I had a typo in your notes. Thank goodness I at least said it the right way every time I said it when I was up here teaching it. But it's written backwards in your notes. This is what your notes should say. This is the way I taught it. It just was written wrong. Okay? Whenever a flagella rotates clockwise, that puts a prokaryote into a random tumble. When a flagella rotates counterclockwise, that puts the prokaryote into a run in whichever direction that prokaryote happens to be facing. Right? So when we did my silly demonstration last time, told you if there was something in the room I wanted to get to and I was a prokaryote, I would have to rotate clockwise to tumble, then stop, rotate counterclockwise, run, stop, rotate clockwise, tumble, stop, rotate counterclockwise, run, and I keep doing that alternating until I ultimately get to the position that I want to get to. Okay? So did everybody make that change for me? Okay? That's important. Okay, we'll flip back to the picture. So that was our flagella. Do eukaryotes have a flagella? Yes, they can, right? But does it look or function anything like the prokaryotic flagella? No, it whips like a tail. Okay? One of the other appendages we went through were fimbriae. Do y'all remember what those are for? That's the little pokey ones. They help it stick into something, right? They help a prokaryotic cell attach. The example I gave you was an E. coli that lives in your intestines, right? He uses those fimbriae to stick into the wall of your intestine. So when poo or whatever's flowing through, it flows and the bacteria is allowed to stay where it wants to be, inside of your intestine. Okay? The last appendage we went over was a pilus. What was it for? Exchanging DNA. Why would bacteria want to exchange DNA with each other? Anything that would help the other one. If I'm a bacteria and I've mutated to be able to live in warmer conditions, well, heck, I'm going to share that with all my buddies around me where we'll have a colony, we'll have a group of us if the temperature gets higher. Antibiotic resistance, anything like that. If it's an advantage to me, then I'm going to share that with everybody else, and that's what we use a pilus for. Why is there no cilia on this picture of a prokaryotic cell? Because prokaryotes can never have cilia. Cilia are appendages you only see in eukaryotic cells. Okay? So I used to not even bother saying it, but then it's like you guys just kind of create it in your mind. It's not anywhere in the notes because there is no such thing as a prokaryote having cilia. Okay? Where we ended the class last time was going through the complex structure of the prokaryotic cell wall. Okay? And we'll flip to that page. So we said the prokaryotic cell wall helps the prokaryote keep its shape. No matter what you do to that prokaryotic cell, it's going to stay the same shape. And there were three basic shapes. What does a coccus shape look like? It's round, it's spherical. What does a bacillus look like? Right, it's elongated. And then what does a spirillum look like? It's twisted up. What does a vibrio look like? Curved comma. And a spirochete. Really, really twisty. We call it a corkscrew. Okay? All right. The cell wall is composed of peptidoglycan. What are the two things that make up peptidoglycan? Protein and sugar. We have a carbohydrate or sugar backbone, meaning that is going to be the basis of this structure. These different colors are alternating sugars. One of them is called N-acetylglucosamine, or NAG. The other is called N-acetylmuramic acid, or NAM. So they're going to alternate, and that forms your basic layers of the peptidoglycan. Holding those layers together is where you find the proteins, and that's what the little blue and green balls are right here in the picture. These are called tetrapeptides. Simply means it's a protein that's only 4 amino acids long. Okay? Just layers of sugar with proteins in the middle. Is everybody comfortable with that? Okay. So we went a little further and we said, yes, all prokaryotic cells, with the exception of a few weird ones that we're not worried about talking about, they have this peptidoglycan in their cell wall. But it turns out there's two different ways we can see this peptidoglycan. And we named them according to what some guy saw about 60 years ago, Dr. Graham. Dr. Graham named the first group gram-positives, and he named the second group gram-negatives. Why did he call the first group gram-positive? What do they have? More peptidoglycan. Plus, plus, plus. Lots of peptidoglycan. He called this one gram-negative because it was lacking all of that peptidoglycan. It does still have it, right? Still got some peptidoglycan. Just doesn't have quite as much. Other distinguishing features of the gram-positive, the gram-positive cell has what we call teichoic acids. These are little negatively charged chemicals sticking out of the peptidoglycan. That makes the gram-positive cell more negatively charged. They're both negatively charged. We saw that in lab. Those of you that had lab this morning, right? All the bacteria were negative, so the positive stains stuck to them. Okay? But the gram-positive is more negatively charged. If we look at the gram-negative has some extra stuff too. It has some extra fat hanging out here on the outside. That is what we call the outer membrane. Now, I didn't really point it out last time because we weren't quite ready for it yet. Notice in this outer membrane, you've got some little guys called porin proteins. What's a pore? What does that word mean? It's an opening, right? Think about it this way. The plasma membrane, which we're about to go through in just a minute, you know that that controls the movement of stuff in and out, right? Well, if I've got another set of a fatty membrane, I'm going to have a way of getting stuff in and out of that too, right? That's what those porins are for. Okay? So since you only have an outer membrane in a gram-negative, you're only going to have porins in a gram-negative. Okay? Now, since they look different, they have different characteristics as far as things that can easily hurt them. The gram-positive, it's a little bit easier to phagocytize a gram-positive cell. Okay? Didn't use that word last time. What does phagocytosis mean? Breaking down of a cell by another cell, eating it, right? Engulfing the cell. So if I'm a big bad immune cell and I want to eat one of these bacteria, it's actually easier for me to grab the gram-positive and eat it. It may be thicker, but the only thing I have to digest to eat this one is peptidoglycan. That's all I have to figure out how to do. So I can break this one down by digesting it a lot easier. If I tried to digest this gram-negative, first I would have to release enzymes to break down fat. Then I'd have to release a new set of enzymes to break down peptidoglycan. Then I'd have to get those out of there to go back to the fat again. So it would be harder for me to digest that one. Even though it's harder to digest a gram-negative, it's easier to mechanically break it. Meaning if I wanted to just pull it apart, excuse me, which does not normally happen. I mean, you don't really grab bacteria and pull them apart. But one thing we do to them is we change their environments, which makes them swell and stretch. Since these are thinner, they're more likely to just break apart. I know they've got lots of different parts, but they're just physically thinner. So they're mechanically going to break apart a lot faster. Okay? I probably shouldn't tell you this, but I'm going to tell you this. You're going to see these pictures on your next test, or your first test, the first one you take with me. So you need to be able to recognize that. You need to be able to recognize the parts, and you need to be able to recognize, of course, which one's gram-positive, which one's gram-negative. So go ahead and know that. What did I tell you was the easy way to tell them apart? The layers, right? This one looks like a hot dog sandwich. Did I see the hot dog sandwich? Right? It's got 4 hot dogs sitting in this gram-negative, and then your fats are your pieces of bread. So that's what I always look for. Okay. All right, any questions about what we did last time? Everybody feels good? Okay. Good, I only spent 10 minutes doing that. All right, so let's do some more new stuff. But it really shouldn't be new. Every one of these terms should be really familiar to you as we go ahead and start talking about the plasma membrane. Okay? The plasma membrane is made of what we call phospholipids. Okay? I'm going to see if my pen will write on my screen. If it doesn't work, I'll give up quick. But if I wanted to draw a phospholipid, I would draw it something like this, right? Sorry it's so messy. It's just kind of hard to tell where the ink's going to come out when you're virtually writing. Okay? But if I drew that, would you have been able to tell me, yeah, that's phospholipids? You know, kind of remember that from biology class? Okay? The reason I draw them is to point out this part of the phospholipid is charged. It's polar. Do y'all know what the word polar means? It can interact with water. Okay? It's hydrophilic. Okay? The entire phospholipid bilayer is not polar. It's not hydrophilic, right? The inside part of it is hydrophobic. It's the fatty part. It doesn't like water. Okay? So can water or a charged molecule just readily move through that phospholipid bilayer? No. Does fat dissolve in water? No. You all know that, right? That's just something we learned in probably middle school science. Fat and water are different. They don't dissolve in each other. We put fatty membranes around our cells, and bacteria do as well, so that things don't just go in and out whenever they feel like it. We're allowed to have some control over that movement. And that is the function of the plasma membrane. It's selectively permeable, meaning it can choose what it wants to move across the membrane, and it can control which way it wants to move across the membrane. Okay? It's extremely important for us, but we're going to look at this from the standpoint of we're a prokaryotic cell and why is this important to them. Okay? If a prokaryotic cell wants to eat, does it have a mouth that it can open to eat? How's it going to do it? It's got to pull it into the body. It's got to absorb it, right? So if it's going to absorb something to eat, it's got to go across the plasma membrane, right? What if it's got a waste product it needs to get rid of? It's got to spit it out. It's got to go across the plasma membrane. So we've got to have a way of looking at movement in both directions. So that brings us to reviewing a few terms that I know you've heard before. Let me get rid of this before I mess something up. Okay? We've got simple diffusion, facilitated diffusion, and osmosis. Those are the three types of passive transport. Passive transport means that we're moving molecules with the concentration gradient. Okay? So how that works. Let's just kind of, so if I just draw a little line right here, okay? And let's say I have 3 molecules on top of the line, 1 molecule below the line. Pretend the line is the plasma membrane, okay? If it's passive transport, which way are those molecules going to move? Down. There's 3 of them on top. That's my high concentration. There's 1 on the bottom. That's my low concentration. So if the X's could move, one of those X's would move down here. That would be passive transport. That means when you're studying this, simple diffusion, facilitated diffusion, and osmosis all move that way, from where you have more to where you have less. So what's the difference between them? Okay. Simple diffusion occurs when you have a small nonpolar molecule. Okay. So think about what we said about the plasma membrane, right? Here's the plasma membrane. The inside of the plasma membrane, it's nonpolar, right? So the only thing that could just move through the fat on its own would be something that was nonpolar. Okay? So let's say I've got 3 nonpolar molecules on the outside. They're very small. And I've got 1 small nonpolar molecule on the inside of the cell. Okay? Since they're nonpolar, they can just move through the membrane on their own. So which way would they move? Right, through the membrane, right? One of the ones on the top that's in excess is going to move down here until we reach equilibrium, right? Until we have equal amounts. This would be an example of simple diffusion. That's as simple as it gets, right? You just got more of them on one side than the other. They can go straight through the membrane, so they even out. That's simple diffusion. Facilitated diffusion, it's just a little bit different. What does the word facilitate mean? Help, right? So it's going to be the same thing. It's going to be diffusion with help. Okay? Facilitated diffusion occurs when the molecules you're trying to move across the membrane are not small and nonpolar. Let's say I have some bigger molecules. Okay? And they need to get through the membrane. They're too big, or they're charged, either way. They can't just go through. They got to have help to get there. So they're going to need, in the middle of the membrane, a transporter protein. That's why when you see membranes drawn, they always have something that looks like these blue things in the pictures up here in them. Those are proteins throughout the membrane. We use those proteins to move things back and forth when we need to. Okay? So it's facilitated diffusion. The facilitator is the protein. It is specifically going to open up only for that one molecule it was designed to allow to move. Okay? Since it's still diffusion, which way would the molecules go? From where I have more down to where I have left. So we understand why they're both diffusion. Both diffusion because they're going from higher to lower. The first one's simple because we go straight through the membrane. The second one is facilitated because we had to have the helper protein. Make sense? I always thought of the little transporter proteins as like a boat. Right? If I wanted to get across the lake, I'm perfectly capable of getting across it, right? But I got to have some help to do it. I need a boat. That's your boat. It gets you across where you need to go. Make sense? Okay. We're going to get to osmosis in just a second. Let's go ahead and talk about what kind of ends up being almost the opposite of passive transport, and that's active transport. Active insinuates the use of what? Energy. So if I have active transport, I would still have to have a transporter protein because I've got to have some way of using energy when I do my movement. It's active transport. With active transport, we are going in the opposite direction of diffusion, of passive transport. With active transport, I'm going to take a molecule from where I have less to where I have more. That's active transport. Okay. I've all learned that before, I'm sure, right? Does it make sense now? I don't care if it made sense before. Does it make sense now? We got it? All right, so let's see if you got it. Let's talk about osmosis. Osmosis is the simple diffusion of water. We all know what simple diffusion is, right? Moving from where you have more to where you have less. Osmosis is just talking about when it's water that's moving in that direction. So we're going to be moving from where we have water, more water, to where we have less water. Okay? So when you did this in biology class, you probably did the example with a red blood cell. That's fine. But it's micro class, right? Let's do it with a bacterial cell. Let's say I have a beaker full of water, okay? And I put some poor little lowly bacteria in there. That's actually a giant bacteria in my little drawing, but so I got some bacteria in that water. Okay? The water, I'm going to put a little salt in the water. Okay? Most bacteria have around 1% salt. Sodium chloride is just salt. So most bacteria have about 1% salt on the inside of them. That's just kind of standard. Okay? Let's say I put him in a solution. We'll do the easy one first. That's 1% sodium chloride. They have equal amounts of salt, right? This is an isotonic solution. Same amount of salt on the inside, same amount of salt on the outside. But I thought we were talking about movement of water, right? So why am I talking about salt? Because if I have 1% salt, what percentage is water? 99%. So if there's 1% salt on the outside, there's 99% water. I have equal amounts of salt. That means I have equal amounts of water. No net water movement. Isotonic. That's what the bacteria want. If you go either direction, more or less salt, that's not good for the bacteria. Okay? So let's say I have a beaker. I'm going to put that same bacteria in it that is 1% sodium chloride. And this time in my water, I'm going to put 5% sodium chloride in my water. Okay? So I'm kind of mistreating the bacteria a little bit. This is a hypertonic solution. I've got more salt outside of my cell than I have inside of my cell. Okay? So the end question is, which way is the water going to go? But before you answer, let me show you my kind of weird way of figuring this out. We know it's going to go from where there's more water to where there's less water, right? So we got to figure out where's there more water. Okay? If there's 1% salt on the inside, what percentage of it's water? 99% water. If it's 5% salt on the outside, what percentage is the water? 95% water. So you all know numbers. Where's there more water? Inside the cell. So the water's going to move from where there's more to where there's less. That water is moving out of that cell. What's going to happen to my cell as the water moves out of it? It's going to shrivel up and eventually die. Okay? Does that make sense? That is the easiest way to think about these osmosis questions. And I don't care how many times I've done it, if I took a test, and you'll see me up here doing it when I'm taking your test with you, making a key. Whatever the question reads, I'll draw my little beaker. And I'll draw my little bacteria in there and put how much salt is in him. I like to visually see it, and that just helps me do it. Okay? So let's go the other direction. I know you'll be able to guess, but let's do a hypotonic solution. So we can keep our same bacteria, 1% salt inside of the cell, and let's put him in a 0.5% salt solution. Okay? Where's there more salt? In the cell. Where's there more water? Outside the cell. So which way would the water go? Into the cell. And what's that cell going to do? He's going to swell until he bursts and dies. So either situation is going to kill that bacterial cell eventually. Do you think we ever do this? You think we use it? Do you think we use the hypertonic way more often? Do we ever put salts and stuff to keep bacteria from growing in it? Every time you open anything that's been canned, it's full of sodium, right? Is that sodium a dietary need? No. That sodium is in that can. That way they don't have to worry if they got bacteria in the can. They just dump enough salt in it. It'll kill whatever bacteria made it into the can. And you may be eating dead bacteria, but it's not going to hurt you. That's what we use the salt for. Okay? It's also the reason bacteria don't grow quite as much in saltwater fish tanks like they do in freshwater fish tanks. It's harder. I know there's a lot of chemical work with a saltwater tank. It's a lot harder to keep a freshwater tank clean because the bacteria grow like crazy in it. Okay? So how do we feel about this? We understand the different movements? You're going to hear the terms throughout the semester. Okay, well, let's move on to something else if you guys have it. Sorry, if this is just a complete review to you, then just enjoy it and make a good grade on the first test, right? Just think of it that way. Don't think, God, I'm bored. Okay? A few things that maybe you may not know from biology class. We're going to now go to the inside of the prokaryotic cell. And most people teach you there's nothing there. There's a lot there. Okay? First off, the cytoplasm. There is cytoplasm. It's very similar to the cytoplasm of a eukaryotic cell, except in the prokaryotic cell, you do not have any cytoskeleton. The cytoskeleton in a eukaryotic cell is made of things like microfilaments, microtubules, intermediate filaments. I don't care if you remember those words or not. But do you remember what a cytoskeleton was for in the eukaryotic cell? Holds everything in place, right? It's like a big scaffolding. It works like your bones do. If you didn't have the cytoskeleton inside of your cells, your organelles, wouldn't stay in place. Well, prokaryotes just don't have that. And that's simply because they don't need it. Because what maintains the shape and the integrity of a prokaryotic cell? The cell wall, right? What we just went over. They have something else to do that, so they don't need a cytoskeleton. Okay? Do prokaryotic cells have DNA? Yes, they're alive, so they must have DNA. That is the way we carry genetic material. The big key here is to understand that prokaryotic cells do not have a nucleus, but their DNA is sequestered into one region, and that region is called the nucleoid. It's not a nucleus, it's a nucleoid. The DNA kind of all wraps and hangs out in one little area, and it is folded nicely, but it's not folded the way eukaryotic DNA is folded. Eukaryotic DNA is folded into chromosomes, right? Little X's you see drawn everywhere when you look in science books. Prokaryotes don't have that. It's still folded, it's still organized, it's just not folded up into chromosomes. Alright, that's not real interesting. What is interesting is that prokaryotic cells have extra DNA. In addition to their DNA they just have hanging out over here in their nucleoid, they have extra DNA called plasmids. Plasmids are not something that bacterial or prokaryotic cell has to have. They house what we call extra genes, things that help them have an advantage to survive. So let's say I've developed antibiotic resistance. I'm going to store those genes on a plasmid. Number one reason is I don't have to have it, so I'm not going to mess with my nucleoid because I don't want to mess up my essential genes. But why else would I want to keep those advantageous genes separate? What am I going to want to do with them? I'm going to want to share them, right? So when we were talking about the pili this time and last time, we said we're exchanging DNA, right? Hopefully at least one of you had the thought, well, how can you pick which DNA and go into this big, long, massive piece of DNA and figure out what to take? You don't have to. Bacteria have figured out, let's just keep those on a little tiny circular piece of DNA out to the side. That way I can replicate it really fast and share it with all my friends whenever I want to. Okay. Now this is, what I'm about to tell you is not something I would ever test you on, but I think some of you will find it interesting. We use these plasmids to our advantage in scientific labs every day. And it is not something that is high-tech or difficult to do. I did it probably once a week when I was working on my degree at USM, right? I mean, that's definitely not an Ivy League high-tech science school, right? What we do with it, we can take bacteria, grow a bunch of them, kill them, purify out just that plasmid. Now I have a little vehicle, a little DNA car that I can cut in half, stick whatever DNA I want into it, and then I can put that back into a new bacteria that's alive. Do bacteria reproduce fast? Yeah. Now I've got my piece of DNA I'm trying to work with in a bacteria. I let those grow, kill them, purify it back out. Now I've got tons of my DNA that I started with just a little bit of. That's really easy to do. Even neater, DNA codes for proteins, right? We learned that, we reviewed that last time we had class. So let's say I take a piece of human DNA and I put it in that bacterial plasmid. I can put it in the bacteria and then tell the bacteria chemically, make protein out of it. And then that bacteria will make my human protein for me, spit it out into whatever they're growing in, and I can just purify my human protein out. Does that sound like science fiction? You know how old that technology is? That is how we used to produce insulin that diabetic patients took. We took the sequence for human insulin protein, the DNA sequence, and we put it in bacteria, and the bacteria just grew and grew and grew and pumped out all the human insulin we needed them to pump out. Very, very neat and very simple to do. All because bacteria have that extra little piece of DNA. So that was just a little extra for you today. I thought some of you may find it interesting. Okay? Helped me get a PhD, so I'm happy about it. All right. Make sense? All right. Next we have ribosomes. What are ribosomes? What happens there? We make proteins. If bacteria have DNA, then they're going to make proteins. So they've got to have a place to make them. Bacteria make proteins just like eukaryotes make proteins. They take their DNA, they turn it into mRNA. The mRNA feeds through the ribosome, produces the protein. Okay? There is a slight difference between prokaryotic and eukaryotic ribosomes. Prokaryotic ribosomes are what we call 70S. Eukaryotic ribosomes are 80S. Okay. The S is just a unit of measurement, like I weigh so many pounds. I could also say I weigh so many kilograms. Right? It's just a unit of measure. 70S is smaller than 80S. Okay? I point that out not because it makes a big difference in function, but once we figured that out, we figured out, hey, we could use this to our advantage. Right? We talked a little bit last time about how a lot of the antibiotics are designed to attach only to peptidoglycan. That way antibiotics will only bind to bacteria. New antibiotics will only bind to a 70S ribosome. So if those antibiotics go into one of your cells, your ribosome is too big, and that antibiotic won't hurt your cell. But if that antibiotic goes into a bacterial cell, it can bind that ribosome, and it's going to kill that cell. Okay? So it's just a new way we make antibiotics. You should learn to know what you're taking. You should never just trust the doctor when he gives you something. Remember, he or she's a human too. Okay? All right. Next organelle, something completely different that is not found in a eukaryotic cell. These are called inclusions. Inclusions are similar to vacuoles. Inclusions store stuff. What makes us not call them vacuoles is that vacuoles always store food in a eukaryotic cell. Inclusions store lots of different things. And one prokaryotic cell can have multiple types of inclusions, but they're all storage sites. The most important one, and yeah, you do need to know the name of it, is called a metachromic inclusion. It stores what we call inorganic phosphate. The chemical symbol for phosphate is P. It's actually PO4, but can you think of anything that's an important molecule in your body, that's going to be in a prokaryote's body as well, that has some P's in it? Think hard. What's an important molecule that has a letter P in it? ATP, right? That's energy. If you need more phosphate to make energy, that's okay. You will eat, again, probably too quickly because we live in the United States. Every time you eat, you ingest more and more phosphate. So when your body needs to make ATP, it's got plenty of phosphate floating around. If that bacteria needs to make some ATP, can he just go eat something when he runs out of phosphate? Okay, of course not. So he has to have a way to store it. When he can get it, he's going to take as much of it as he can in by active transport. He's going to store it. Polysaccharide granules store glycogen and starch. Those are complex forms of sugars. Why do they need to store sugar? What is sugar the starting process of? What do we do with it? Energy production, right? It starts with glucose. That's their way of storing energy sources. What now? Well, I would say, I almost said that actually, but then it dawned on me. They store fat too. They store those in lipid inclusions. But just like some people store more fat than other people, some bacteria store more fat than other people, right? Some of them prefer to store fat. Some of them prefer to store sugar. Okay? Humans store glycogen too. We do store sugar. Okay? So it's actually kind of an exercise myth that if you exercise enough, you will automatically start using fat. You won't. First, you're going to use glycogen. You're going to use your stored sugar before you use your stored fat. Okay? All right, the last four are some weird ones. So you don't know as much about them, but I think some of you may find some of them a little bit interesting. Sulfur granules store sulfur. There are some bacteria that live exclusively off of sulfur and not oxygen. They are the bacteria that produce horrific odors. Have any of you ever been to Yellowstone? I have not. You ever been to Yellowstone to see the geysers? Well, the geysers are, you see a picture of it, it looks gorgeous. It's this, huge thing that will erupt with really hot water, and it comes every few minutes. I don't know how often, but it's on a time schedule. It's always going to erupt. But when you go there, it's hard to even look at it because the whole time you've got your hand over your nose because you don't want to have to smell what's around it. The bacteria that live in the geysers live off of sulfur. So it smells like they're like passing gas the entire time. It smells like rotten eggs the whole time you're there. Now, I've never been, but that's what they say. Carboxysomes, I won't talk about those. Those are really complex. It has a lot to do with photosynthesis. But the last two are kind of neat. Gas vacuoles store air. It gives a prokaryotic cell buoyancy. If you're in the water swimming and you want to swim lower, you just turn down and swim down. If you want to swim up, you swim up. Fish work the same way, right? If a bacteria wants to change where he is, he doesn't have any appendages to swim in the water, and you say, oh, he's got flagella. They don't work like that in the water like they do in some other environments. A lot of those will just change the amount of air they have on the inside of their body. If I'm in the water, or even in the air, if I let more air out of my body, which way am I going to go? I'm going to sink, right? If I suck air in, I'm going to float. So they can do that. They can adjust where they are in water or in air simply by passing gas or pulling gas in. The magnetosomes store iron oxide. That is a magnet. Okay? We really don't know why bacteria, they're rare, but why some bacteria have magnets in them. But what we do know is if you have them under a microscope and you put a magnet on one side of the microscope, they'll all turn and look. Look, they're not looking. But they'll all turn and face the same orientation. If you move the magnet to the other side, they'll all slowly get there and they'll all face the other direction. Okay, maybe that doesn't sound neat, but think about it this way. Our only explanation we could possibly have is maybe these bacteria have figured out how to know whether they're facing north, south, east, or west. Isn't that how a compass works, right? It points north based off of the magnetic core of the Earth. So we think that these bacteria may have figured out how to tell which direction they're facing. That's pretty smart for one cell to be able to do. And we just think that's how they did it. We don't know for sure, but that's what we think. Our last organelle is called a endospore. You may as well get used to hearing this word. You're going to hear me say it in lab with disgust quite often. I said it already this morning. And you're going to hear me say it a lot when we get to diseases. Endospores are made when a bacterial cell is in some sort of environment that is going to be a danger to him. The bacterial cell will produce an endospore. The endospore can survive even if the bacterial cell dies. And then when the conditions get better, that endospore can become a new bacteria. It's a way to give them survival. It's not reproductive because one cell makes one endospore becomes one cell. So you're not amplifying the amount you have. It's just allowing them to live pretty much no matter what you do to them. This is why I get so mad at them in lab. Because I put them in the autoclave, I sterilize the media, then I give you the media and I say, here's your media. And I put staphylococcus in it. Look for it. And then you show me on the microscope and I say, I don't see any staphylococcus. All I see is bacillus. It's because the bacillus that are always in your cultures in lab, they can make an endospore. So every time I put them in the autoclave, they just turn into an endospore. And then as soon as they come out of the autoclave, they pop back out and start growing in my cultures again. So they're just kind of everywhere. They're extremely hard to kill. There are ways to kill them, but it's not easy to do. We have found endospores buried in glaciers. This is the dumb part. We've brought them into scientific labs, and we have allowed them to germinate to see what they were. And that's how we've discovered some ancient bacteria. If you ever watch the Sci-Fi Channel, like that's the way the world will end. You ever seen Andromeda Strain and things like that? Okay. So it's really dumb that we did that. But that just proves these things are really, really hard to kill. A lot of our really nasty diseases we're going to talk about are caused by endospores. There's only 3 genera, 3 different genuses, if you want me to say it wrong, that can make endospores. But that's enough to make some nasty diseases. Bacillus anthracis, that's the bacteria that produces the endospore that causes anthrax. All you have to do is inhale 1 Bacillus anthracis endospore, and you can develop pulmonary anthrax. The mortality rate of pulmonary anthrax is 100%. That's a pretty high percentage if you ask me, right? All because you inhaled 1 endospore. Clostridium is another genus. Clostridium perfringens. If that gets into an open wound, that causes gas gangrene. And then your body will begin to just rot in those places, and you end up with having amputations and just really nasty situations going on. All because an endospore got in there. Clostridium tetani causes tetanus. Okay? You guys don't know what tetanus is, probably, because you've all been vaccinated. Let me tell you what tetanus is. Tetanus is not lockjaw. That's just what we think it is because we are very privileged Americans. Okay? When you get tetanus, all you have to do is get 1 endospore into your body. The first thing that happens to you is your muscles begin to contract uncontrollably, and you get twitches, uncontrollable twitches. If you keep it and you do not get medication for it, it leaves your body into convulsions. Convulsions that make your body bend forward 90 degrees, backwards 90 degrees, forward 90 degrees, until they paralyze backwards. So you lay there in an arched back, paralyzed position until the muscles around your lungs paralyze and you suffocate to death. That's what tetanus is. Okay? Sounds great, right? All because of an endospore. Guess where that endospore lives? In the dirt. We could go dig up some dirt outside right now and I guarantee you we would find one. When's the last time you had a tetanus shot? Has it been soon enough? If you don't know the answer, guess what you need? You need a tetanus shot. Every 10 years you need a tetanus shot. Okay? All right. So I'm not trying to scare you. I just think that's amazing that that one little spore can cause horrific diseases that can do those things to you. Okay? So let's look at how it's made. So here's our little bacterial cell. What shape is this? Bacillus. Not rod. Bacillus. Gotta wake y'all up every now and then. Okay? So the first thing that's going to happen, he copies his DNA. Because if he's going to make some sort of structure that he can use for survival, what's the only thing he's really got to put in it? His DNA. That's the only thing he's got to protect. So he copies his DNA, starts kind of sequestering it off here to the side. Then he starts making membranes and putting membranes around it. The membranes that he puts around his DNA are made of dipiclinic acid and calcium. I know you don't know what dipiclinic acid is. You don't need to. But you know what calcium is, right? What organs in your body have calcium in them? Bones and teeth. Are those hard structures? Yeah, that's why we put calcium in them. Make them hard. So this bacteria basically makes a bony structure around his DNA that will help him survive whatever conditions are going on. He eventually spits out that what's now going to be the endospore, the membranes wrapped around his DNA, and that endospore can live indefinitely until situations get better. Then he can germinate and become that, just like that original cell again. Okay. So really, really, really neat little guys. This is one of those things that is 100% unique to prokaryotes. Nothing like this in a eukaryotic cell. Okay. Now we're back to the eukaryotic cell, and we went over this last time, right? Are there any questions about this? If not, we got one more thing we're going to do together. We understand what each organelle does in the eukaryotic cell? Okay. The last thing I want to tell you is, I want to kind of give you a story, and it is a theory. A theory is a scientifically accepted, meaning proven hypothesis. Okay? So this is not something you can say, well, I don't believe that. Well, that's fine. You don't have to believe it. But in order for it to be a theory, it has to have scientific evidence backing it up. The endosymbiotic theory tells us how eukaryotic cells developed. It has been scientifically shown that the most likely way a eukaryotic cell was formed was that first we had prokaryotic cells. Why do we think they came first? Some of the parts of them are simpler, but mainly because some of the oldest prokaryotic cells can live in extreme environments. In the very, very early days of Earth was an extreme environment. Volcanic eruptions, all that good stuff. So the only thing that can live in volcanic lava is a prokaryotic cell. We have very low levels of oxygen, high levels of nitrogen, high levels of sulfur. Only thing that can live in those high levels of nitrogen and sulfur, prokaryotic cells. Okay? So that's why we state that only prokaryotic cells could have been there at first. Then they think somewhere along the way, one prokaryotic cell somehow ingested another prokaryotic cell. So now you would have one cell with another one inside of it. And that this could have evolved where the ingested prokaryotic cell became the nucleus. Okay? Did that happen overnight? Of course not. It would have taken a long time. So what's the evidence for that other than, well, prokaryotes probably came first? You have DNA inside of your cell in places other than your nucleus. Inside of your mitochondria, you have DNA. Every plant inside of his chloroplast, he has DNA. And it's different than the DNA inside of the nucleus. So if we're following this theory, well, if another prokaryotic cell got in there, that could have eventually have become another organelle. And that's what the endosymbiotic theory tells us, is that prokaryotic cells came first, eukaryotic cells came second. Okay? As far as fitting this into religion, that's going to depend on your religious beliefs. I'm a Christian, and I'm a scientist. And I could, if anybody ever wants to sit down, I could tell you how it all fits perfectly together. But it's a much longer discussion than what you guys really want to sit here and listen to me explain to you. Okay? If you want to hear it comes... Right. Well, I mean, the two easy answers is this could have happened before God, or God, or whoever you believe is the creator of all things. Who's not to say he's the one that made this happen? So you could look at it either way. But I find that kind of stuff really interesting. So I'd be glad to sit down and have a discussion with anybody that wants to. But I'm probably not technically supposed to do it standing up here in front of class. I don't want to lose my job. Okay? All right. So let me tell you what this is. At the end of each of your PowerPoint notes, you have this. This is not really homework you have to do. Where's your homework you have to do at? On Blackboard, online on Blackboard, okay? This is where I went through the questions at the end of the chapters in your textbook and just told you which ones you should probably look at. The reason you don't have to do it is because you have the answers. The questions and the answers are in your textbook. So it'd be silly for me to assign you something that you already have the answers to. But as you're studying for tests, it would be a good idea to read the question and the answer, and just a way to help you study. Okay?